In this session, we'll be speaking about the antigen processing and the antigen presenting. Right? So, as you all know, we started speaking about the antigen processes. Before going in detail regarding what is antigen processes, I think you should know the meaning of each. Right? Right? You know the meaning of antigen? Right? Right? Now let's know the understanding. Let's understand the meaning of processes actually. Right? A simple way, the definition is antigen process is an immunological process that prepares antigen, right? That prepares antigen. This is antigen, right? This is antigen presenting cells. This is T cells. Just observe when I read. Antigen processes is an immunological process. Yes, it is an immunological process that prepares antigen for presentation to specialized cells to the immune system called T lymphocytes. It is considered to be a stage of antigen processing. That means, in the simple way, in the local language, let's understand that process is nothing but the moment the antigen enters in our body. The moment antigen enters in our body, antigen don't have a separate flight to attach to the T cells. It doesn't have any flight to go near the T cells. It has to go on the certain basis of, you have got something like an antigen presenting cells. It takes the antigen the moment it comes, it processes, it breaks it out, and then it wraps it in a gift and it gives to T cells. It's not something like the antigen, the moment it enters in our body, it doesn't goes inside or doesn't meet directly to T cells. You have to meet the broker first. You have to meet these antigen presenting cells, and the antigen presenting cells, it's gonna change the modification or change the process. That means it's gonna do the processing of this antigen. Process in the sense it's gonna do something to the antigen in the antigen presenting cells like macrophages, and then it present to the T cells. And now let us understand that it's written that next point, it's written like thymus dependent. What is thymus dependent? Means T cell dependent. That means now we are understanding that how the T cell, how the T helper cells is activated. Right now T helper cell is here. Right now antigen is here. Right, you understand that antigens are of two types. One is endogenous, one other is exogenous. So one is endogenous and next is exogenous. And what is endogenous? Right, these are the proteins. A transformation occurs. They act as an antigen in abnormal condition like in oncogenes, tumor cells or viral infected cells. I'll teach you everything in detail, but right now understand that this is a cell. Right, this cell is infected with the virus particles. Right, this cell is infected with a viral particle, and now the viral particle in the nucleus, in the cell, and now it is inside the cell. So I have called it endogenous. So and endogenous is an abnormal conditions like in oncogenesis or tumor cells or viral infected cells. You got some abnormal proteins in the cell, and that protein or abnormal cell or the virus which is present in the cell, it has to be kicked out. Right, we have to kill this out. Right, now that is all about the endogenous antigen, what's written in our notes. And what is exogenous? Exogenous is nothing but which is coming from outside in the sense, if this is the cell, right, if this is the cell and this is the cell, antigen is not right now in the cell, it is coming from outside. It might be bacteria or some another antigen which is coming from outside. So we are calling it exogenous antigen. Right, so that is the difference between the endogenous antigen, and that is the difference between the endogenous and exogenous. So that's all about the endogenous, what's written in our notes, and that's all about exogenous, what's in our notes. And now let us understand the proper way of how this endogenous antigen which is present in the cell is given to T cell. Right, how the antigen which is present in the cell, how it is presented to this beautiful T cell. Right, before going in mechanism, you should know some things regarding this, and that is MHC. Right, I already discussed in detail regarding the MHC1, but now let us once again understand what's written here. So Just understand that. Let me read what's present in that and explain you in detail. So it's written that all nucleated cells, I'm speaking about the MHC1. So to all those guys who are confused with MHC, and just think that right now this is a cell. Whenever antigen is presented, now antigen is inside the cell, right, inside the cell, and it is presented to the T cells. That means the moment the antigen is inside, these MHC particles take this antigen, and through one strand to one protein, this is an antigen here, right? Now this is known as MHC. MHC is something like something which is presenting the antigen to the T cells. It's like a hand, right? And this is like a gift, right? And this is like a hand. So this is known as MHC. And MHC are of two types, MHC1, MHC2. And now we are studying about this MHC1. MHC1, what's written in our notes, it is present in all nucleated cells and platelet contains, right? The platelets and all nucleated cells contain this MHC1, right? And it's also written that there is something called as peptide binding group for MHC1 can accommodate 8 to 10 amino acid length peptides, very small molecules. A peptide binding group for MHC2 is 13 to 18 amino acid length peptide. And it's also written that MHC2 doesn't play a role in endogenous antigen. MHC1 plays important role in endogenous antigen for antigen processing. And what is this? What is it talking about? What are these guys? So now let us understand each line from the notes in detail, right? So let us go with this point of view. So it's written that first point, the MHC, this is an MHC molecule, is present in all nucleated cells. That means it can be platelets, it can be epithelial cells. On the epithelial cells, you got some, in the epithelial cells you have these MHC molecules. It is present in all nucleated cells and platelet contains. So right now you understand, it might be confusing. You have got something like MH2 also, but this MHC is present, MH2 is present in antigen presenting cells. But right now you understand that the first line is it is present in all nucleated cells, all nucleated. That means right now we understood that this MHC1 is present in the nucleated cells, somewhere in the cells. And now let us understand the next point. It's written that peptide binding group for MHC1. MHC1 can accommodate 8 to 10 amino acid length peptide, a very small molecule. What does it mean? Suppose if this is the cell of any nucleated and this is MHC, let me cut this section and draw it beautifully here, and this is what it's written here. This is one of the nucleated cell. On the cell membrane you got a protein known as MHC1. This MHC1 contains alpha 1, alpha 2, alpha 3 and beta microglobulin structure. And what are you seeing? The grooves, right? This is a groove, this is known as groove, and this is known as peptide binding groove, right? This is known as peptide binding groove. What is meant by peptide? Peptide is nothing but something within protein molecule. From where did we get something known as antigen, which was found in the cell, endogenous, right? So that is bound here. And now how much large can it accommodate? It can accommodate a small, it just of 8 to 10 amino acid, right? It is just an 8 to 10 amino acid length of this peptide. It can accommodate that point, right? But whereas you have got MHC2 structure, if you see the MHC2 which is present on the antigen presenting cells like macrophages, if you see the structure guys, you have got an alpha 1, beta 2 and alpha 2, and you can see the groove here, and this peptide groove can accommodate 13 to 18 amino acid length. So that's what it's written in your notes. The peptide binding group for MH2 is 13 to 18, whereas the peptide binding group for MHC can accommodate 8 to 10 amino acid length, a very small molecule that was. And now understand that MHC2 doesn't play a role in endogenous antigen. That means if some antigen is present inside, to get it to present to the T cells, the MH2 doesn't play any role. It is the MHC1 plays an important role in presenting the antigen which is present in the cell to the T cell, right? And now let us understand where is it present. It is found in the inner aspect of endoplasmic reticulum associated with Golgi complex, right? That means now we know that till here we knew that and this MHC is present, right? These MHCs are present in the cell. Where exactly in the cell? The exactly in the sense you have got in the inner aspect of, and we'll be dealing in detail, but right now you understand that MHC is present in the endoplasmic reticulum, right, in association with Golgi complex. That means with he is associated with Golgi complex. And right now I think you have understood everything what's written in our notes, right? And now let us understand the concept, the mechanism of this endogenous antigen, right? The mechanism of endogenous antigen, if you read this, now let me read, then I'll explain you in detail. The first thing written in our notes here is, right, the endogenous antigen enter into proteosomes, break down into small molecule, enter into tap and passes to MHC1 passes through Golgi complex and enter on antigen surface. Then T cytotoxic cells kill them. Then you have what is TAP, it is transporter for antigen presentation. Then these you have got a syndromes, one Bayer lymphocyte syndrome and Bayer lymphocyte 2 syndrome, and all these TAP proteins, all these we have to understand. Now let us understand the concept, and reach each line of the DAMS notes in detail once again, right? Let me show you what did I tell you. Endogenous is nothing but you have got an antigen, suppose a virus which is present here, and now let me teach you how this virus which is present here is presented to the T cells. So let us go, right? Are you ready? Yes, let's start the action, right? 1 2 3 4 action, right? See guys, this is a cell, right? This is a cell, this is a viral infected cell. Which infected? Viral infected. And this is that virus. This is so dangerous here, and this is something like, right, this is viral cell, right? This is viral cell, so dangerous. This virus, this endogenous antigen, this is a virus which is present in the cell, and now it's time, because it's gonna kill our cell. I think we have to kill it out. And why? Then something gonna happen in the cell, right? So let's see the first step. Let's correlate with our notes. The first step, it's written that the endogenous antigen, right, this is nothing but, and this is a proteosomes. What is proteosome? Proteosome is nothing but it breaks the proteins. It's gonna break the proteins, and this peptide, this endogenous antigen, it enters the red one, this is known as proteosomes, right? This is known as proteosomes, right? As you all know, we started speaking about how this endogenous antigen which is present in the cell is kicked out. Now right now in the cell you got the step you have to understand here. This antigen enters, yes, it enters into the, right? And what's written in our notes? These proteosomes break down into small molecules, enter into a tap and pass to MHC. What does it mean? It means that you have got an antigen here, it has passed through this proteosome. Through the proteosomes, you can see the long peptide has broken down into small peptides, a small one, and the peptides which are present in the cytoplasm of the cell, now they are entering into the endoplasmic reticulum through a protein, through a antigen, that is TAP. This is TAP. What is TAP? TAP is nothing but transporter for antigen presentation, right? So right now we understood the peptides which is present in the cell is broken down with these proteosomes. Once it has been broken down with the help of these proteosomes, the long peptide bond has broken into small peptide bonds, and these small peptide bonds enters into this endoplasmic reticulum. They enter the endoplasmic reticulum through this TAP. What is TAP? TAP is nothing but it is a transporter for antigen presentation. What is the function of this TAP? It takes the protein, a small molecules which is present in the cytoplasm, to endoplasmic reticulum, right? And next point, what's written in our notes is MHC1 passes through Golgi complex, and right now the antigen is bound to this MHC1, and these are the things what you can see. The red one is, those are MHC1 molecules which are present in the endoplasmic reticulum, and these endoplasmic reticulum, these MHC1 are bounded to the small parts of these proteins which have entered the endoplasmic reticulum through this TAP. from the endoplasmic reticulum. From the endoplasmic reticulum, now it goes into Golgi complex, and you can see this MHC1 with this small peptide, right? You can see the small peptide with this MHC1. And now this MHC moves through a vesicle and it goes on the cell and tells the T cells, dude, I got antigen, dude, you have to do something. Yes, you have to do something. I got an antigen here, right? And this is how the process had happened, right? This is how the process has happened. The first, I repeat once again, we started speaking about the endogenous antigen processing. You got an endogenous antigen, say example, you got a virus which is present in the cell, and this virus enters through these proteosomes, and this viral particle is broken down into small proteins, and the small proteins which is present in the cytoplasm enter into the endoplasmic reticulum through a TEAP tab. What is the tab? Is nothing but a gate known as transporter for antigen presentation. What is the function of this tab? It moves the antigen which is present in the cytoplasm to this endoplasmic reticulum. And now from the endoplasmic reticulum, in the endoplasmic reticulum you have got MHC1, and these proteins which have entered into the endoplasmic reticulum, they go and bind on these sites on the MHC1. I have told here, this is the MHC molecule which is present right now here, and this is the peptide bonding site, and this antigen is bound right now to here, right now they are with this association with this MHC1, and later this MHC1 goes through this Golgi complex, and from the complex it expresses antigen on its surface. This is how we expressed. Right now you should understand something. There are some syndromes, guys. What are the syndromes? If there is any deficiency of this tap, right, if the tap is not working, what is tap? Not water tap. What is tap? Tap is the transporter for antigen presentation. If if there is a deficiency of this tap, it leads to no MHC1 expression. How that means? If there is no tap, now there is no movement of the antigen which is present in the cytosol to endoplasmic reticulum. If there is no movement, now there is no connection of the antigen to the endoplasmic reticulum. If there is no binding of this, then there is no forming and there is no binding at the membrane point. Right, so this is known as a Bare lymphocyte syndrome. What is a Bare lymphocyte syndrome? Bare lymphocyte syndrome is a deficiency of this tap leading to no MHC1 expressions. Yes, if this tap is closed, then the antigens do not enter into endoplasmic reticulum, then it doesn't bind and there is no such MHC1 on the membrane. Right, so and now you should understand that this tap protein, right, this is a tap protein, breaks a large endogenous antigen molecule into small molecule, and this MHC1 with endogenous antigen transport to the Golgi apparatus. I have told you, right, with this, and MHC1 can't express antigen. Okay, if remember that this is a normal cell, this is a nucleated cell, and now let's see this interaction. Right, you got an interaction, the movement. Right now this is an antigen, right, this is a cell, right, this is a cell. We have got an antigen here, endogenous antigen. Right now it has expressed, right now it has expressed. The moment it has expressed, now it is waiting for someone. For whom is it waiting? It is waiting for someone. And who is that guy who is waiting? And who is none another than T helper, means CD8 cell. It is a cytotoxic cell. The moment it interacts with the T cell receptors, right, and it's gonna kill this. Right, we'll be dealing how it's gonna kill, but right now you understand that now it has bound. Right, now it has bound here, and now it is a cytotoxic cells. And just time being, you understand that you have got perforins and granzymes. These perforins break this membrane, the granzymes enters and it kills these cells, and now there is no more cell because this is killed by cytotoxic cells. So that's what it's written in our book. If MHC can't express antigen, NK cells will kill. So I have to tell you that I didn't tell you right now. So I spoke about the TAP protein break down large endogenous antigens to into small molecules. MHC1 endogenous transport to Golgi complex. Okay, I have told this. So now let us understand that. Right, now let us understand. Right, So what does it mean? If MHC1 can't express antigen, NK cells will kill. If NK cells can't kill them, tumor cell develop. If endogenous antigen bind to MHC1, then it is autoimmune disease. So what is he trying to tell? Okay, so he's trying to tell that, guys, this is a normal cell. On the normal cell we have got MHC1, right? And this is something known as activating ligand. Okay, and on NK cells, natural killer cells, on that also you got some receptors known as inhibitor receptors and activating receptors. These activating receptors always it try to activate this activating ligand, so that this receptor always it try to kill. Okay, so but we have this MHC1, when it binds to this inhibitor receptor, it controls so that the NK cells recognize that yes, this is our cell because we have got MHC1 right now, so this NK cell doesn't kill. But what happens in some conditions, if there is no expression of MHC on the normal cells, right? If there is no expression of MHC on the normal cells, then what happens, you can see the excess stimulation of this activating receptor on this ligand. Now there is no control of MHC, right? There is no control in the sense, now inhibitory receptor, there is no inhibitory receptors now. Why there is no inhibitory receptor? There is no MHC on few cells. Like you have gotten, suppose you have got a tumor cells or viral infected cell, they'll be like, if there are no MHC, right, in the normal cells or any cells, if there are no MHC on them, then NK cells, what does it do? It goes and thinks that yes, this is an foreign, it's not our body, and it start releasing perforins, granzymes, and it kills it. Okay, that is what he was telling. If MHC1 can't express antigen, NK cells will kill. If NK cells can't kill them, so if NK cells can't kill them, right, if the NK cells can't kill them, so this is a tumor cell, right? Because this is a tumor, it's not expressing anything. This is a viral infected cell, right? If it is a tumor cell, if it's not expressing MH cell, normally it has to be killed by these NK cells. Suppose if it is not killed, right? If it's not killed, then the tumor starts growing, right? Tumor starts growing. So that's what he was telling, that if not killed, then tumor cells develops. And let us understand the last point, guys. The last one is, if endogenous antigen bind to MHC2, then it is autoimmune disease. And normally in the cell, right, in the cell, endogenous cell, it has to bind to the MHC1 only, right, and it should be present. Suppose if this endogenous bind to MHC1, right, then it is autoimmune disease. That means it simply tells that this is an endogenous molecule, it should bind with this MHC1. Suppose in case you even have MS2 also, right? Suppose if there are MS2 cells, if there are is telling that if this endogenous cells, instead of binding to MHC1, if they bind to, I'll tell you what is MS2 in the further session, but right now it's written that if endogenous antigen binds to MS2, not 1 is 9, normal, it's fine. If it binds to MS2, then it is autoimmune disease. Then it is autoimmune disease. We'll be dealing about what is MS2 in all that. But right now this is all about the endogenous cells, guys. So hope you have understood here. So I think you have got no confusion regarding each point of view. So that's right, this is what it is, right? So that is all for now. Let's take 2 minutes break and let us continue with this exogenous antigens, right? Thank you, guys.